meeting, as you've noticed. So this meeting is henceforth being recorded. I was going to just say that I'm really happy to be here because I feel it's an important synergy that we get from Amsterdam and Rotterdam. I mean, like I'm really delighted to have been introduced to George in the context of this collaboration. The paper that we published was about a very cool hypothesis, I think, where we had basically, long story short, the cerebellum can control coherence between sensory and motor cortices. So it makes sense. I mean, the cerebellum works as a glue, and this is one of the papers in which we publish, I think, one of the most compelling evidence that this also happens dynamically. And through a model from Georgia, no less. I mean, despite the fact that I am a modeler and this is my hypothesis in the paper, just for you to have an idea how, like we modelers, we also particularize and specialize. And so certain models are very adequate to answer some certain questions. And if you already have the model, sometimes you can deploy them across the board. So I had imagined a model that was similar to Georgie, but making the model is a different question. And so it was a beautiful fact that George's model could immediately be applied, or mutatis mutandis, applied to our context, and in fact reproduce the results that we observe in reality, I think, to a very compelling way. So very nice to, for you to think in the context of modeling. There are a vast variety of models out there. Each model has its own role to play. So even, it's not one modeler fits all. Okay. And today I'm going to be telling you a story on modeling on Hodgkin-Huxley type neurons, which is slightly different than the Wilson-Cohen models that Georgia used himself. And you might have probably, most likely, have already heard about the Hodgkin-Huxley models, I'm presuming, since now this is the part of your course in which you talk to other scientists about their computational work. And so you probably already have the basics. And this is an incredible benefit for me as a lecturer today, because I can skip a lot of details and get to the juicy bits. This book, whose page I am using as this word synergy, because I thought it was a good time for us to toast together, by Mark Latash, is If You Are Interested in Motor Control, is a book that I recommend with my full heart. There are many stories out there about forward models and inverse models in the brain. This is not the only game in town. And I think that the arguments against certain simplifications that some of the modern winners, the dogmas, are having can be interestingly debated, to say the least. And I would then like to point you to this nice book. I'm not going to talk about synergy today. This is in the context of motor synergies, right? So multiple muscles and muscle-skeletal synergies and how motor control happens. But I just thought it was a good opportunity for me to let you know that this exists. I arrived in Rotterdam from Japan, and in Japan I was working with Erik de Schutter, who is a computational neuroscience working on the modeling of the Purkinje cell and cerebellum, because it is so potentially one of the it was the mecca of the cerebellum, Rotterdam, when I was searching for a postdoc. And I met Chris DeZeo. So here we've discovered, for instance, the zebrin bands from the cerebellum that was Jan Vogt in the context of Rotterdam. And they had like an enormous lab that concentrates on cerebellar aspects, and it's an immense amount of knowledge we got from that. So I went then from Japan to Rotterdam to study the cerebellum because I thought that we were getting close. And what brought me to the cerebellum was the internet. So the internet makes the cerebellum appear particularly amusing in many respects. So these are all kinds of non-functional motor behaviors that require quite a bit of explanation in many of these cases, including motor coordination. And so cerebellum is a very funny system, right? I mean, so these are the kinds of motor actions that we expect the cerebellum to make. I don't know how well Zoom is getting across with these animated GIFs, but here you have postural corrections, vestibular reflexes, optokinetic reflexes, visomotor coordination, and on-the-fly corrections. It's all motor corrections as being cerebellar function. Fairly complex in the case of the bottom right there. Also, another thing that we shouldn't forget is that most motion is on the basis of some sort of oscillations, and oscillatory behavior is a very fundamental thing we also have to explain, and even its dysfunction. I mean, it's very interesting to see how sometimes global brain dynamics involves even the entire body, especially during development. From motor babbling to, and so motor babbling is a lot of oscillations. I'm happy to have now my second daughter. She's now 1 month old, or my second child, a daughter, and it's the most delightful thing to see, the kind of interesting oscillatory motor babbling behaviors that they produce. So cerebellum is concerned with motor movement. Obviously, you know this already by now, I'm sure. But you probably have also heard that the cerebellum works on the basis of errors and motor errors. So this is you waking up in a corona day, in a quarantine in the end of 2020. And so I wanted to point out by using this example that motor errors are not necessarily a very well-defined concept. I mean, as you saw the guy pole jumping and this cat here, it's a lot about it's not necessarily about minimizing the distance between the desired target and its trajectory, but it's a more complex thing that involves the entire essence. So in this case, you have an automatized behavior, and the automatized behavior is interrupted by a surprise because expectations fail to obtain. But this is not a well-determined three-dimensional targeting problem. So it's a more complex problem than that. And the kinds of things that we have to then explain when we're using oscillatory behavior are very functional, especially in the case of humans. Of course, that's what we want to know. How is it that we learn new, completely fresh ways of interacting with the world and potentially producing sensory motor loops and feedbacks such as this? You have the control of a bow. So this is Valentino Breitenberg. If you haven't heard about Valentino Breitenberg before, that's another one of my literary recommendations in the context of cerebellar explanation. This is an amazing piece called The Cerebellum and the Physics of Movement, where he discusses the necessities of motor control from the perspective of muscle contraction. So in these three cases, he is actually comparing the three types of bowing here, détaché, martelé, and staccato, and you see that certain protocols, despite the fact that there are three oscillatory behaviors, they will require substantially different control protocols. And if we think about the control problem according to the constraints of the kinematic constraints, kinematic functional constraints, because you also want a particular sound to be coming from your violin. If you contextualize motor behavior in a more functional, holistic frame than simply looking into trajectories and the distances between targets and desired points. So here we have the findings of the agonist and the antagonist. I mean, so these X and V, this is the position of the arm, the velocity of the arm, the acceleration of the arm, and then this is the firings of the agonists and the antagonists, as we presume to be observing. So to start with, I'm going to tell you a couple of aspects of olivocerebellar anatomy and physiology that you may or may not know, but I feel important to take into account when we're looking into an explanation of this system. The reason why I am So the talk today is about a particular model of the inferior olive, of the olivocerebellar system. But I think that there are more aspects of anatomy that demand an explanation in the context of the olivocerebellar system that do not get the attention they deserve. Now, you may know that the cerebellum and the cortex, also through the mediodencephalic junction, converge onto the inferior olive. So you may know that that there are Purkinje cells, like in the cerebellum in green, that project the cerebellar nucleus in blue, that then inhibit the inferior olive. And you may know that the inferior olive is sending back climbing fibers to the cerebellar Purkinje cells. But what you may not have known is that when you unfold the entire cerebellar cortex, it is very different than what you get when you unfold the cerebral cortex. So here you have on the left a cut of the cerebellum, and you see its neat involutions. If you what happens if you unfold it? And it unfolds a little bit like a napkin. I mean, you can actually create a cerebellar model with a napkin. I've done so to much amusement during dinners with other scientists. And the thing is that it's much longer than it's wide. The cerebral cortex, it unfolds as a disc. The cerebellar cortex unfolds as a very, very long toilet paper folded. Now here you see in scale a bunch of different cerebella from different animals, and that have been unfolded according to the procedure here on the left bottom. Now the challenge here is find the human. And now if you would, if you can also try to find the mouse or the monkey or the cow, but how about this? Go into the chat and vote. Which one is the human? Wow, Georgie, nice. Okay. Yeah, 14 seems to be the winner. Very good. We do have a 15 and a 12. So here's another funny story before I tell you the solution to the riddle. Valentino Breitenberg, brain anatomist, medical doctor, conducted an entire talk on human brain anatomy by showing other anatomists the brain of a cow. And the question is, can you tell the difference? And apparently no one was able to, during his talk, like his very persuasive speaker, to point out that that was not a human brain. 15 is the cow. So 15 is long but thin. 14 is the human. The main expansion, and in fact, it's the main growth of any cortex out there, together with the cerebral cortex, was the lateral expansion of the cerebellum, which is related to tool use and sensory motor behaviors. It's the part of the brain that we study when we're looking into the whiskers of the mouse. So here we have like a lateral expansion that in the human has related to potentially to tool use. So the two things to think about now here are, you are an anatomist in the 19th century or 20th, and you're looking at unfolded cerebellum and you're wondering very hard, why the hell? Why the hell? Why is it long and not circular like the rest of the brain, right? Leave this to you. I mean, it's a lovely question for us to discuss later, because no one actually knows right now. I mean, there are a couple of suggestions, and part of the suggestions have to do about the modularization of the functional output of the cerebellum. But there is no final solution to why long and not wide. But it is an invariant in anatomy, and therefore something very interesting to talk about. Now, one more piece of that particular puzzle. The cerebellum Purkinje cells receive from, and the cerebellar cortex receives from cells in the inferior olive here, where you have this little red arrow here. And Sugihara and friends, they reconstructed entire traces from the climbing fiber from the inferior olive into the cerebellum. And what they found out is that cells very close by, they actually map along this vertical dimension here. So in a very tiny little space of the inferior olive, you talk to an entire vertical stripe of the cerebellum. Colors represent individual cells reconstructed. So you see that, for instance, in red here in lobule 7 in the cerebellum, one cell broadcasts that information across the entire convolution there. Fun, huh? I mean, like, if you're not marveling at that particular circuit, I don't know how I can excite you about the cerebellum. The cerebellum is assembled in a loop. I had actually mentioned this earlier quickly before, but this is a more diagrammatic way to show it. And the point that I want to make is about, there are two. One is about convergence. So these climbing fibers from the inferior olive, they go and they meet for one inferior olive can talk to up to 10 Purkinje cells, but one Purkinje cell receives from exclusively one climbing fiber. Now you have a number of these Purkinje cells, and I'll tell you in a moment what their physiology looks like. And they have some inhibitory interneurons here. The BS is the basket cell, it's inhibiting Purkinje cells. And these Purkinje cells are converging onto the cerebellar nucleus. Converging, right? So 40 Purkinje cells to one cerebellar nuclear cell. From the outside, you have these parallel fibers, and so the mossy fibers are coming from the rest of the brain and are conveying all of the information, essentially, to the cerebellum via the parallel fibers. If you remember the arrangement between Purkinje cells and parallel fibers, if you didn't see them before, I'll show you a Purkinje cell, and I'll tell you now that the parallel fibers are perpendicular to the Purkinje cells, and the Purkinje cells are flat. So the stimulus coming from the entire brain is distributed across the Purkinje cell arbors, which are flat via the parallel fibers. The connection from the Purkinje cells onto the cerebellar nucleus is is inhibitory, as you saw here in the key. And the cerebellar nucleus to the inferior olive is inhibitory. So this is a bi-inhibitory loop. Now, keep this in mind. The inhibition that inhibits inhibition when I continue the story. Here you have the fact that the cerebellum of the human and the primate has been shown to have particular functional zones that relate to particular parts of the body. And these functional zones in the cerebellum are related to functional zones in the inferior olive. Today I'll show you a reconstruction of the mao here on the bottom left of it, which is the medio-accessory olive. And it is related to the vermis, which is the main center part of the cerebellum here in blue. And that one is known to correlate with eye motion and torsional movements and postural corrections. So, and talks to the fastigial nucleus in the cerebellum. So when Purkinje cells, now let's talk a little bit about physiology. You've seen spikes, and many of you are coming from artificial intelligence, and part of the thing that I want to impart on you is the heterogeneity of the brain signals with respect to what they're doing, so that you can potentially find new analogies to create new machine learning techniques that try to capture them. Now here, the olivary neuron is on top. It fires very few spikes at about 1 hertz. And it has these subthreshold oscillations that you see here indicated in orange. That olivary neuron produces the climbing fiber signal that arrives into the Purkinje cell and produces a complex spike. And the complex spike is this spike going upwards here in this B Purkinje cell trace at about 1 hertz. And that one pauses the simple spike train that was coming. So the simple spike train is a spontaneously produced spike of the Purkinje cell that comes at about, from 50 to 200 hertz. And when the complex spike comes, simple spikes pause. So you're thinking about the disinhibitory loop. So when complex spike from the olive comes to the cerebellum, it tells the cerebellum to stop. So the Purkinje cell stops. The cerebellar nucleus gets disinhibited, so it fires. And when it fires, it inhibits the inferior olive, right? So this is the loop. And you can see here that there's a little burst in the cerebellar nuclear neuron. I notice now that there is one here. Sorry. This was not supposed to disappear. The idea here is that this complex spike, when it arrives at the cerebellum, it produces an immense calcium influx. And so this is a model of the calcium influx by the arriving from the climbing fiber onto the Purkinje cell, where you can see in D and E the calcium concentration, and in A, B, C, this are the voltage during the complex spike. So voltage is brought up, calcium enters the cell, and this calcium is the one plasticity, driver signal for the plasticity mechanisms of the Purkinje cell. There are two, as in most cells. So you can either strengthen the connections between the parallel fibers and the Purkinje cells, or weaken them. If you don't see complex spikes for a long time, the synapses that have been just used are strengthened. If you just saw a complex spike, the synapses that are just used weaken. So now you have basically most of the machinery to try to understand what the dynamics will look like in the entire cerebellum. And before I go further, I just wanted to show you a little video with an experiment in my lab where we recorded simultaneous Purkinje cells. Simultaneous Purkinje cells here in the trace of yellow and in the trace of blue. The stars on top, they indicate complex spikes, and you can also see that despite the fact that these Purkinje cells have been recorded about 300 micrometers away from each other in crus, you sometimes get, for instance, synchronous pauses. Here, there's a pause here in yellow, and there was a pause in the yellow simple spike trace, and also a pause in the blue spike trace. It just so happens that in this case there wasn't a complex spike here, but the blue complex spike came here and then creates a pause. The red and the yellow bars here, they indicate the momentary firing rate at the center of this of the video. So the two sounds are from the two different complex spikes. And this gives you a sense of the cerebellar activity. So this is the sound of the mouse licking. And now you have a little bit more data. Now, I'm not going to tell you a lot about this particular experiment, but what I'm going to point out to you is that something was missing during licking. Which was like complex spikes were not coming. If I play it again, you will definitely notice the two of them came in different times. Now, they were coming most of the rest of the time. They're very spontaneous. I mean, they come like relatively frequently, and the funny thing is, there was no obvious error for you to associate it with, despite the fact that they even came simultaneously sometimes. The complex spike is a pain on the butt of every cerebellar neuroscientist out there. And because they're trying to make sense of these complex characteristics of the complex spike. The complex spike is the spike that appears in the Purkinje cell that has been originated in the inferior olive, and that is going to occupy the rest of my talk now. So, they're powerful. There's one climbing fiber per Purkinje cell, and there's thousands of synapses on a single Purkinje cell from a single climbing fiber to make sure that there's going to be calcium distributed in the entire arbor. They are complex in shape. They're not simple on and off spikes. They have like humps and bumps, and they can be long and short. And we have published a paper where we observed complex spikes with calcium transients of up to 30 milliseconds. So they're weird with respect to their dynamics. They're unreliable. They have very low frequency. They have an enormous broad latency to stimulus. And I'm going to show you some data now that obtained in our lab. They're also multimodal. In the sense that they, everything can cause them. Multimodal is a fun way to say that we don't know where the stuff is actually coming from. It comes from vision, comes from audition, comes from tactile, comes from smell, So. And they're weirdly synchronous, and they're synchronous also spontaneously. I'll show you data to that. And they're super important because they drive plasticity, as Masao Ito had determined in the 1970s and had been predicted priorly by David Marr, who by now I presume you may have heard from. So so here's another quiz, just so that I orient myself with my crowd. How many of you have heard about David Marr before? Marr, M-A-R-R. Okay, so I'm going to type his name here and say it was a genius working on understanding the brain. He was one of the guys that talked about segmentation earliest. So he, talked about disparity between the two eyes and saying that can use the information to create object recognition. The way in which we atomize brain function cognitively these days is often using a Marr like perspectives, so the three levels of Marr. So he says that there is a construction level, then there's an algorithmic level, and there's a functional level. Very crucial guy to learn about because he was one of the giants on whose shoulders we stand on, especially in modern computational neuroscience that is trying to converge with neural computation. Here's some data. This is a two-photon image of about 500 micrometers of cerebellar cortex of the mouse. And in red, you will see the calcium signals coming from the Purkinje cells. You see lines, like horizontal lines. These lines are basically the Purkinje cell arbor from above. The Purkinje cell is flat, and when you look at the cerebellar cortex, you see Purkinje cells on top of each other. Now, what comes clear in this piece of data here, is actually real-time recording, is the incredible synchronicity of these complex spikes. They come in large patches and they come really synchronous. When you look into the traces, like luminance traces of these complex spikes, so here we have in one field of view, say about 20 different Purkinje cells that have been recorded, and we looked into their calcium-related signal, which is a luminance trace. Now, notice here, this is my lovely collaborator, Lawrence Bosman, and he decided to mark two cells in red so that he would show you when they come synchronous. Like he was saying, oh, look, there's cross-correlation between two cells. It's not only between two cells, it's between the entire set of cells, right? So the 20 cells that we were recording at one point had produced a complex spike. This is not a very selective signal for learning. Moreover, so like I'm just pointing out that the Lawrence is thinking about, there are two cells that fire together. No, there's all of the cells are firing together. This is the mindset we should be having when we're looking into the cerebellum. Why are they firing together like that? And what causes the synchronicity with respect to non-synchronicity? If you can see my cursor, you will notice that in some other moments you have a smaller subset of these cells firing synchronously. Also, you notice that the time basis of this is very long, 10 seconds, and so these cells, these spikes are coming very infrequently, as I mentioned. So, funny thing. Go do an experiment. What causes the complex spikes? And then so you go and you start to measure these calcium traces. And here on the bottom, you have the dendrites in a field of view. From cells 1 to 26. And before you start to apply a stimulus in blue, they're already firing synchronous. But once you start to apply the stimulus, then they're scrambled. Yay! So this happens with a variety of stimuli. We've done so we've published this recently, in which we tested all types of modalities. Another very diligent work by Lawrence and others. So in here we tested a bunch of different modalities, like tactile modalities, LED, and we are looking into what causes the complex spike to modulate. And the complex spike doesn't really like stimulus that much. I mean, it's not like it's being evoked by a sensory stimulus most directly. And you can see here in a vast variety of these cells, unless you produce an air puff to the face of the mouse. If you produce an air puff to the face of the mouse, then the number of complex spikes across the entire region grows immensely, and you have, oh look, I have my event-related potential. No, I have the event-related spikes, and they're more synchronous. So here, what we're showing with these stacked histogram bars are the spikes produced for different Purkinje cells at the same time. So you see here where my cursor lies, that there was many cells that fire simultaneously at the production of the air puff, but not so much when we've poked the lower lip. So an intense stimulus is going to evoke synchrony, that's for sure. I mean, like if you slap the mouse in the face, he's going to wake up from his slumber in the setup and produce a bunch of complex spikes. But they were coming anyway, and they were coming synchronously anyway. So very careful now. Funny, we've tried to punch these cells on the face and saying, okay, but let's do differently. Let's try to see if they're in training to a certain rhythm. They have subthreshold oscillations. I'm going to tell you more about this in a moment. Have subthreshold oscillations. We bump them on their face with different frequencies, and we see whether they're increasing their firing rate. Now, this is the most glorious negative result I have ever obtained. So we've created air puff frequencies onto the face of the mouse as a trivial experiment. And then we measured complex spikes in Purkinje cells very diligently. And here what you see is the delta in frequency for stimulation frequencies in X. So at 1 hertz, at 2 hertz, 3 hertz, 4 hertz. So we would expect that if these are sensory driven events like this, I just showed you, oh look, I mean there's an air puff here that I produce, and there's synchronous spikes to my air puff, but then it doesn't happen every time that I puff it. So, obviously sometimes it happens and then it comes synchronously, but the gain increase in firing frequency is virtually inexistent. So this is the wild type and the connexin 36 knockout on the right. But this doesn't matter. I mean, both of these animals here, respectively from their spontaneous firing frequency, which is about 1 hertz, famously, they don't modulate. That's weird. We have a model that shows why, a couple of reasons why this could be the case. But if you want to know more about that, then you read this paper, because that's not what I'm going to be talking about today. I just want to tell you that it's weird. When you look into sensory evoked Purkinje cell spiking, we might make incorrect inferences. So this is the interim conclusion for the experiments that I've shown to you so far. The complex spike are very synchronous. They are promoted by input correlation via sensory input, but they do not seem driven by it, this synchrony. And there's a very low firing frequency and very underwhelming firing frequency modulation. So these are kinds of elements that we want to be able to integrate in any model that we make of this system. More data. I don't know if you're in for the data here, and there will be a model, and I hope that you will be pleased by it too. But I want to tell you one more thing.
Could I ask a question real quick?
Absolutely, please.
So as for the synchrony, you would expect that either the neurons, the Purkinje cells themselves, or the climbing fiber, so the actual neurons in the inferior olive, are highly interconnected, so forming a sort of block that fires together or not. But is that the case?
Yes, that's the case. And this is what the entire talk about, like the entire model is going to be about.
Oh, sorry.
Great. Thank you for bringing it up. So, and I'm going to tell you the reasons why they're firing together, but more importantly, when do they fire together? Because the point now is that they're not always firing together, and they're not always firing to stimulus. So the questions would be, when do they fire together? Which fire together? And how do you cluster them up? What is the meaning of them firing together? So 1955, Golgi and Nissel staining of the inferior olive gave us a lot of data about how these connections look like. And there's a couple of things here but I only want to point out the bottom left plot. Here, Scheibel and Scheibel shown in the 3-day monkey how the inferior olive cells looked like. So they're curly. They're close. In A is one subnucleus. Some of these cells are very curly and very local, which would make sense. I mean, they connect to their neighbors, as Nicole just pointed out. But you also have these long-range connections in B. So these actually coexist in the same nucleus. He's just indicating them in the diagram separately. We've recently redone these experiments with much more attention to detail. And here we've reconstructed the zillions of these neurons from the inferior olive and entire olive to see whether there were particular morphologies in particular places, and to try to get a sense for what is the relationship between these curlies and straight neurons. The straight neurons go very far, and the curly neurons are close by. So how does the anatomy of the entire nucleus looks like? Nora Freeler, she actually also studied it through. And Mariuka, my collaborator in OIST. So we measured a lot of stuff about these cells. And you can see here that their dendrites remain very close in certain cases. In the bottom, you have total dendritic length of these cells. And so you can see that some of the cells are crumpled up, but their dendritic length is more or less the same as some cells that are not crumpled up, so on the right here. So this is a fairly directional cell that's reaching a much farther place. So we've measured the huge amount of these characteristics and tried to find what were the morphological correlates of these metrics. So we measured the stems and diameter and so forth and so on to find that indeed there are certain things like curly cells and straight cells, as had been observed in 1955, but they are not as clearly separate as commonly happens in biology. It just so happens that it appears, at least from the PCA perspective, that they are living in a morphological continuum. And so here you see this little sphere and transforming into a cone. That is how eccentric the cells are, and PC, the first principal component, indicates the amount of straightness of the cell. You can see here in the correlate between principal component 1 and the straightness. Curly cells make the majority of them. Straight cells, fewer in general. But here we have a balanced sample, right? So in principle, we had annotated the cells according to what we believed that the cell class that the neurons had. So we thought it was like some of them were really very curly, some of them were really very straight, and some were a bit ambiguous to our experimenters' eyes. And yet, we were able to reconstitute this idea of straightness, but only along a morphological continuum. Then you ask, okay, but how are these curly cells organized? Back to the point of Nicole, how are they connected together? So these are 3 arrangements that we find when we're looking into the cells that participate in the same cluster. So in the bottom, in the top here, you see that certain cells have their dendritic fields that are going towards each other. They're eccentric, very eccentric. The soma is at the rim of the dendritic field. But sometimes they are also eccentric, but pointing in different directions, as in B. And often what you can see is that multiple cells converge onto a Gordian knot, or onto a little bit of a cluster-like dendritic set of fields. Only that you have some very long cells and very short cells. And this is the architecture that now we want to understand in terms of the dynamics. So it would seem as if by doing these reconstructions, we find cells that are in fact not only curly, but in clusters, and they effectively avoid each other, whereas other cells cross the clusters, producing what we believe is something like multi-scale connectivity. So this is a cell that has dendritic fields from both clusters at the same time, but it doesn't make a synapse. So that's a funny thing. I mean, like, even though it sends its dendrite very close to another cluster, it doesn't make a contact, and that's why it's green here. Okay. So picture is validated. We have clusters, but sometimes even close by cells are not connected. And so what's the scheme and what does it mean? Do a reconstruction based on the positions of these cells. These anatomists, my friends, they had annotated in the slice every single position in XYZ coordinates of every single soma of the nucleus here in green. So the edges. So the vertices here will represent cells. I'll show you a moment, another picture of this in a moment, actually, maybe. And this is just to tell you that, quickly, we've reconstructed connectivity on the basis of different schemes of possible connection. And what we find out is that most likely these cells produce clusters, or most cells, these produce high cluster coefficients if they are connected within 3 connection radiuses of each other, which would be about 60 to 80 micrometers. So we find this plot here. I'm not going to explain detail. We just try to reconstruct a bunch of these connectivities and measure the clustering coefficients as a function of connection radius. The point here is that the reconstruction that we obtain is topologically more similar to a 2-dimensional plane than to a 3-dimensional volume. So nuclei in the brain, they're often arranged in 3-dimensional structures. They're blobs in volumes. It's cortices, like the cerebral cortex, cerebellar cortex, they're flat. And here we show that the inferior olive, even though it's a nucleus and it's inside the brain, it's also flat. That's important topologically. Oh, and now we say, okay, 1050, give me the modeling, give me the modeling. Well, there's a bit of cerebellum for you. It's fun stuff, I hope. But here, I'm going to calculate some of the dynamical consequences through a model. So we know these properties of the inferior olive. They have intrinsic subthreshold oscillations, these cells, 1/3 of them, via certain mechanisms. They have electrical synapses, so they're connected to each other via cytosol, gap junctions. Stop me if you don't know. They produce these high threshold calcium spikes that are very infrequent with multiple traces, and they produce this, the complex spikes. And they produce coherent background oscillations because of the gap junctions. Now these are properties that we had already encountered in experiment before. For instance, here in this experiment by Rodolfo Linas and Elena Lesnik, they applied a gap junction blocker, and they've shown that the global coherence in a slice disappeared at the presence of gap junction blockers, even though the subthreshold oscillations remain. So here in the top control, you have the red being the global signal, global activity signal via voltage dies, and the black is the intrinsic, the pipette recording, the intracellular recording of this individual cell. So you see that the cell continues to oscillate, but once you apply the gap junction blocker, it no longer oscillates with the entire network. So it's no longer coherent. we do a reconstruction of the nucleus with the cells we have. So we take the positions of those cells and we apply to each of them a 3-compartment model, Hodgkin-Huxley type, that has the ion channel mechanisms that produce subthreshold oscillations and high threshold spikes. So each of the vertices here is a cell, and the edges represent reconstructed connectivity according to certain rules, in this case simply proximity. And these channel dynamics will then produce a subthreshold oscillation. So this is how the model looks like. So you have gap junctions from across the cells, the connected cell 1 and 2. Then you have these different currents, the IH, calcium activated potassium, SK type, high threshold calcium, and so leak of the dendrite, calcium low threshold, and I'm sure that you probably all got very bored every time that you listen to all of these ion channel bestiary. They're important. And in fact, this is where we're actually missing things in our artificial intelligence mindsets. We're often not representing any dynamics, or trivially. So the nice thing about this model is that it had been very neatly debugged with respect to its dynamical systems properties. And by now, I think that you may have encountered a bifurcation diagram before to show where spiking is coming from, and to show also how particular parameters, such as the conductance values of the individual currents, impact on the different types of firing of these cells. So we took the reconstruction from our friends in Okinawa, and we put to each single vertex a little model that has 3 compartments, dendrite, soma, and axon, where we have the parameter space that gives us information about what is the type of firing that these cells produce. And here you have in A, B, C, D on the left, the different protocols and different types of baseline firing of these cells. So cells that are quiescent, and if you hyperpolarize, they spike. Cells that are producing subthreshold oscillation in B, and if you produce a stimulus, hyperpolarization, will create a little burst of spikes, and so forth and so on. And we understand very well, and this is this is what's important for this kind of modeling, to understand their bifurcation scenarios here on the right side, to see how they transition from one type of phenomenological spiking type to another. If you spread these, if you spread the conductance values randomly a bit about certain mean, you can obtain a sample of these types of firing. So here I created 64 cells by randomizing their properties. The nice thing is that these manifolds are fairly smooth, and so you obtain, despite the fact that you're changing many parameters at the same time, here I'm randomizing most of the parameters, you have a selection of cells that can be, 1/3 is quiet, 1/3 is spiking, 1/3 is oscillating, and so forth and so on. Then you apply these dynamics of the individual cells to the entire reconstructed network. Okay, so I'm going to first show you what happens if we produce, now we have the network, and I distribute these cells, 1,000 cells in this case, for an entire reconstructed medio accessory olive, the MAO of the inferior olive. And, without further ado, this is the activation protocols that you observe from them. On the left, you have each soma indicating the potential of each cell going up and down. Is in millivolts, the color here on the left. And on the right is the phase transform, the Hilbert transform of the membrane potential values, which gives us the phase, so from 0 to 2 pi, or 0 to 1 cycle. And what you can see here is that in the bridge architecture that I just showed you above, you have the cluster-like activity from multiple different little groups that fire together, even though, or The subterrestrial oscillations are coherent, even though their membrane potential oscillations can be very small. Notice that on the left side, there's very little amplitude in most of these cells. They are oscillating like with a couple of millivolts only. So this is, if you get a strong AMPA, they're probably going to spike anyways. So this is the mindset to have, is the fact that despite the fact that the clusters are coherent in phase, there is wide variation in the amplitude of these cells, meaning some cells are driving this. First thing we've observed, when we have a architecture that has Curlies and bridge cells, the bridge cells, obviously, because they're connecting across different clusters, they will mix together the oscillations from the different clusters and produce some harmonics. So on top here, we have an experiment in which we've have all of the networks connected, both with Curly cells, 90% of the cells, and 10% bridge cells. And the colored cell is the cell connecting different clusters. And you see that that cell produces harmonics. If you disconnect completely the cell from the network, the entire dynamics of the connected clusters will change, and they will reduce their oscillation, obviously their coherence, the long-range coherence. Here's another example in which when we have a bridge connected, it's not producing intrinsic oscillations, but it's producing these harmonics at low frequencies. If you disconnect that bridge, it doesn't oscillate. So this is what the connectivity then brings in from the mixing of these clusters together. Now the fun arrives from the fact that this was a prediction that we had from data that we had recorded in vitro from intracellular recordings. Here on the left, we have 18 or a set of, I think, maybe 16 cells, 17 and 18 are not showing for some reason, in which you see these kinds of harmonics. And you can then look into the spectrograms of these cells and see that some of the cells are actually changing their frequencies. They have multiple frequencies, one embedding in the other. You also see some funky behaviors or typical bifurcation type behaviors. So this cell number 4 here at one point produces a spike and stops firing. Maybe could be two things. Either the cell died or it will start oscillating again in the future. And in fact, I can tell you that I didn't put the data in, but it will start again oscillating in the future. Still mean that it's dying, but it also means that the bifurcation structure recapitulates that of the model. And you have these oscillations in different frequencies and so forth and so on. So this is fun because now we can then look into the intra-cluster and extra-cluster cells and see whether the probability of the harmonics is larger in the bridge cells. And we have data that will, goes well along showing that direction. So this is the first thing I wanted to show. Then we have, we produce hypotheses about the architecture. We said because of these Curlies and bridge cells, because these Curlies and straight cells, different schemes would have substantially different dynamics. So we can now compare the dynamics between these different schemes. It could be, like Nicolas suggested earlier, well, it's simply cells that are close by together, fire together. In fact, the entire network is firing together. I mean, if you connect them locally like this, as you see in a homogeneous or isotropic way, then most likely the cell is going to engage, without a perturbation, engage in a coherent oscillation. If you, however, have a bridge cluster architecture where you have locally connected cells and globally connected cells in smaller proportion, you have multi-scale in anatomy and what happens in dynamics. Or you could have, for example, simply clusters. I mean, cells that are connected to to their nearest neighbors and their nearest neighbors only. Bit of measurement metrics here. So this is, we use spectral clustering to help us evaluate the consequences of each of the architectures that we've picked up in terms of the statistics of joint firing, because ultimately that's what we can relate to experiment, right? So how often do we see synchronously firing cells? What is the scope of synchronously firing cells in the cerebellum? What is the connectivity that we need to have in the olive in order for a certain breakdown to be the case? I mean, say, have multiple clusters, say 50 clusters will give you 50 groups of cells that can fire together. And so if you measure all the cells in the cerebellum, you could potentially distinguish these groups. And in fact, we can do this via statistics. So spectral clustering we do on the Hilbert transform of the membrane potentials of these cells. This is the pseudocode. I will provide you the slide, but I'll just give you a sense to how we're doing this in the via a couple of pictures. So ultimately, what we do is we take in each of the cells, we take the membrane potential of each cell, and we produce the partial correlation of these, and we try to figure out which of these are most likely most similar. And we do this via the graph Laplacian of this partial correlation. We create, from the graph Laplacian, we actually use a k-means for different hypotheses, for different possible numbers of clusters, compare it with Bootstrap, and we obtain in the end a metric that's called cluster stability, where we hypothesize the number of clusters and see what is the likelihood that two cells will belong to the same cluster given a different number of clusters. So for each number of clusters you produce a k-means based on the eigenvalues of the graph Laplacian. Say, for example, take the graph Laplacian, take the first, for one cluster you take the main eigenvalue, and then obviously everybody's going to be belonging into the same cluster. You do this for two eigenvalues, and then what are the largest eigenvalues? 2, what are the cells that belong to the same cluster? 3, 4, 5, up to 120 here, because we have 1,000 cells, so it would make sense. And in this particular case, in this architecture, with this particular stimulation, we found out that most likely the network has something like 30 clusters. And when we organize the cells, like the correlation matrix as above, according to the clusters that we hypothesize, you notice that, well, I mean, it does a fair job of bringing them together. Say, for instance, here, cluster number 3 going from top to bottom seems to collect very well cells that are firing very closely together. It's about 10, 15 cells that are firing very close together. So this block is very clean. There's no more, appearance of other cells that look similar and so forth. But this other bigger block here is a bit more contentious. There are a couple of cells that are similar to cells in cluster 5, but also similar to cells in this bottom cluster here far away.
Could I ask a question?
Yeah, please. Yeah.
So does this come from your model data or from?
It's from the model data. It's from the model data.
But don't you already know the number of clusters?
Very good question. We know the number of anatomical clusters that we put in.
And this is different from the measurements that you get?
Yes, indeed.
Okay.
So we select the number of, so we can do this for obviously different numbers of clusters that we assume. And we can also do this for the homogeneous connectivity in which there are no clusters.
Mm-hmm.
And I'll show you in a moment what looks in the case of the continuous connectivity. Because the question is, say in the continuous connectivity, is it like just one big cluster and that's it? If you produce cells that have different intrinsic activities, they will connect themselves closer to their neighbors. If you've ever played with a Kuramoto model, you know that the distribution of intrinsic frequencies will determine which cells are going to play more likely with each other. So even though we might have a completely homogeneous connectivity, the dynamical property of clustering is different than the anatomical clustering property. So thank you for the questions. Instrumental time. I should have a slide for this to pause now and ponder. Because are the anatomical clusters equal to the dynamical clusters? The point that I'm making is that they're not. And the point that I'm making is that depending on what I will be making is that depending on the type of stimuli, you will observe different parts of this network firing together. Recapitulating what we saw when we had the two-photon imaging data from synchrony, in which you had some moments all of the cells firing together, some moments only a fewer group of these cells. So there's a dynamical organization here that is the relating to the clustering property. So in this case, I put actually 50 clusters inside the network, and 30 appear. Means some of them merged together from the original ones that I had.
A really cool result.
And what I like, and it's going to be nice, I think, in a moment. So this is what the membrane potential traces look like after you remove them. So they look compelling, even though you have substantial variation in the wave shape, right? So like here in cluster 19, you can see that this 19, so the yellow neuron is trailing most of the other neurons. It has a fairly different profile. It probably has a much larger membrane potential than the other, so this is normalized so that we can put them all in the same one. You can see their similarity, but it's phase locked. So even though one of them is probably driving the rest, it's coming after after the entire group, and it feels very consistent. So these particular clusters that I selected to show you, it feels like the algorithm did a fantastic job. And you can also see that cluster 12 and cluster 1 are at different. They're anti-phase of each other. That's kind of cute as well. And so, but there are some moments in which this picture is less clear, and because it's not less interesting because it's less clear. So here is the membrane potential of the oscillatory cells on the left against time. So I have 700 oscillatory cells. I display here the membrane potential, blue and yellow. So yellow is high, blue is low, doesn't matter. But what you should be able to see here is that some interesting phenomena. For instance, here in the first arrow on the left, we have what appears to be a cluster that is together and then detaches. Right, so it's coherent over the first second, and becomes incoherent or breaks down in two groups. You can also see a little bit in the correlation matrix because they stop to be so correlated. If I had only 1 second of data, they would be perfectly correlated and the cluster would be very clean. In the second one, it might actually be a different cluster in the second. Then you have something like this, the second arrow. Cells are incoherent and then they get coherent. This is very, very exciting. And then in the third group, in the third arrow, they're perfectly incoherent. I'm going to throw out a word out there. In 2017, physicists have determined that chimera states can happen in coupled oscillatory systems existing in reality. Chimera states were predicted, theoretically, but never observed. Until 2017, people saw it. What are chimera states? Precisely this. When you have weakly connected networks where certain subgroups can be coherent in certain times and incoherent in other times. So you can have the coexistence of coherent and incoherent states. This is cool because if you now produce a stimulus, then the way in which you're selecting cells is very, very particular. And they still have their subthreshold oscillations. They still have their places where they're most likely to fire, because if they're very depolarized, they're most likely to fire if you get a stimulus. But you have certain groups that are slaves of a certain coherent rhythm, where other groups will actually be more atomized, like in more pieces. Fun thing about this is that you have a lovely way of responding not only to something that you've been in training. So think about the motor system. So we are producing we're producing oscillations in the muscle to keep tension, and we're producing those activities like in EMG, electromyographies, and those are going to come in as feedbacks through the entire system. They are coming into the cerebellum, also into the inferior eyelid, potentially promoting certain particular coherent schemes. So you have a way to trace what are the different phases of your muscle activities, and potentially keeping those phases at different in different moments. So this is the slide in which I discuss what are the motor system implications of these assumptions. We haven't talked about what happens at the cerebellum, but remember, these complex spikes that are appearing from the system, they're driving plasticity at the Purkinje cell. So, groups, right? So we know that in cerebellum there are certain anatomical modules, but we didn't know how they're dynamically allocated during motor movement. When you're doing a coordinated motion, what you would like is to create broad synchrony, but not across all of the motor outputs. If you have 700 independent synergies, or 120 as people have computed, 700 different muscles, but 120 different synergies, you would like to bring sets of muscles together at different times and computing disturbances according to these coherent subtraction oscillations. So we've compared the stability, the clustering properties for these different architectures, and we see that for the bridge stability, you have this tracing tail that is neither homogeneous. I mean, it makes sense, right? Ultimately, it's neither homogeneous connectivity, where the most likely clusterization is at very few clusters, 1 or 2, to a little bit flatter, to cluster only, in which basically many types of clusterizations have stability. This has to do with the fact that you can create in a bridge type architecture, in a multi-scale type architecture, you can create either large groups or smaller groups, whereas in clusters, all of the groups are atomized, like they're all equally likely. If you stimulate the system, also interesting things happen. Let me show you this graph here. At 3,000 seconds, there is an AMPA stimulus that comes in the middle of the network. And let me see, actually, so they're not in the same time basis. In the left, you have the membrane potential for these two. Let me try to play this again. Focus on the phase on the right side. When the stimulus comes, in the Curlin bridges architectures, you get a, I don't know if it's possible to see, and I'll have to plot this in another way to put it on paper. But what happens is that when you get the stimulus on a purely clusterized architecture on top, obviously the stimulus stays there where those clusters that received them were, and the other clusters are incoherent. In the bottom, suddenly all the clusters are brought together, and there is a little bit of a wave propagation, which can be observed as systematic delays in the Purkinje cell, which we have observed in data. And like, you know, this is the stuff that I can't show you everything that I have. Like, I'm already like 15 minutes over time, but I wanted to give you a sense for where we go with this data and how we ask these questions. So we had data, we had anatomy, we had physiology, our big explananda are both at the level of statistics of firing and at functional behavior for the entire cerebellar loop. So this is then for the stimulated case, and you have the same recapitulations of what I said before. Clearly, the cluster stability of the bridge is going to be something between the completely clusterized architecture and the homogeneous architecture. But the nice thing is that only in the bridge architecture can you create coherent groups away from each other when you have simultaneous stimulus, and then you can keep them coherent because you have the bridges, because you have multi-scale connectivity. Summary. Curly and straight cells are a morphological continuum, support multi-scale connectivity, at least as we observe in the mouse. The inferior olivary cells produce these regular oscillations, which are very neat, up and downs with calcium T, and I didn't tell you the mechanisms, but we know them. And wacky oscillations, which are these harmonics. And the harmonics are very likely due to bringing multiple oscillators together. The cluster membership is a dynamical property, which is cool. This is what I think is cool about this result. Even though I put anatomical clusters there, the multi-scale clustering is likely driven by the stimulus, or in fact context. Doesn't need to be stimulus, could be context. And we predict that there will be chimera states in the system. We can then use this data to then correlate with the statistics of the real brain. Stuff to be done is to correlate this at a large scale in a functional scenario. We got recently a nice fat human brain project grant for this little project of mine called Arborio. If you didn't know, this is risotto type rice. IO stands for inferior olive, and it's a bunch of Italians. So collaborating with the University of Pavia and the Institute for Advanced Studies in Trieste, with also people developing this new simulation language for biophysical models called Arbor, which I support. I mean, like a neuron is great, but it is 50 years old. You know, you're better off programming an assembler. So Arborio is a more modern view in large-scale modeling of compartmental cells. And these are friends and fellas, and there will never be enough space for everybody. So thank you for listening very much. And you did ask a couple of questions, and they were instrumental. So thank you for helping me.
Thank you very much, Mario, for the wonderful talk.
Yeah,
We can have a cool discussion. Thank you.
Yes, we have time for more questions. You guys have more?
Yeah, I do. Can you
Yes,
maybe revisit what the implications are for the motor system behavior?
Yeah, I actually have one of my students here. So Elias Santoro, maybe he could tell us.
Hello.
Yeah, I mean, so I'm going to answer. Do I have your slides, Elias, from recent times? Okay, but it's a loop, right? So olivary cells are connected to Purkinje cells, and when complex spike comes, they train the Purkinje cells to whatever context they had. So the recently used spikes, recently produced spikes in I/O, will correlate with the recent inputs in the parallel fibers and LTD them if complex spike comes in particular moment. So obviously this can look into co-occurrences or time stimulus, temporal stimulus. If you have a certain state in the body, you are producing some corollary discharges in the cerebellum and in the inferior olive. When you produce this stimulus, something, I mean, like, you know, a postural disturbance, you're creating, can I read you, like, when we're talking about this? So I'll read you this quote, since you ask, and then I'll tell you. So in the most general terms, the idea that may be drawn from observation outside, right, Aristotelic. The cerebellum checks on global aspects of motor behavior, especially on the mechanical disturbances propagated passively throughout the animal's body. It is reasoned that the sequences of activation of various muscles should respect the phase of the mechanical waves which are set up by external disturbances. You're creating these temporal correlations and you're putting those those oscillators in phase with certain stimuli. When you have a closed loop, you have resonances. Long story short, what we observe is that the system with cerebellar plasticity starts preferring stimulus that comes at the Purkinje cell with particular frequencies. So if you have mixture of inputs coming into the Purkinje cell, you will select certain frequencies in certain Purkinje cells as a function of their closed loop. How exactly that happens, you will have to get a talk by Elias and he will tell you. Yeah, and so we can also discuss more. But because of this closed loop, there is a delayed feedback onto the Purkinje cell. And so the Purkinje cell receives the outcome of what it done and trains those synapses, but spontaneously. One of the things here is like error-driven learning will tell you that the cerebellum produces error spikes via the climbing fibers. And I tell you, no, the climbing fibers are doing spontaneous activity. So what we most likely will be seeing here is unsupervised learning of kinematic features of the musculoskeletal system. This is what the cerebellum is doing. It is organizing kinematics in the loop, in this resonant loop. And so what these olivary groups will do, I mean, they will also create correlations across multiple Purkinje cells, and as you saw before in the anatomy. And so this is why I present the anatomy, is so that I can bind now in the conclusions. Right? So if you have here, say, a functional anatomy, and a certain set of muscles, or a certain synergy more like, is encoded in, say, lobule 7 of the vermis. It will come into this area of the fastigial nucleus. It will come into this area of the medial accessory olive. But stimulus in the body never come by themselves. They come, you know, you fall if you stumble. I mean, like, you know, you move two arms at the same time. And the ways in which you sequence those corrective actions have to bind across multiple muscle groups and create these resonances, and both in closed loops and in open loops, in which, you know, stretching your arms or stretching your arms forward will also make you let's see, if you stumble, you will stretch your arms forward. Some of these are reflexes, so allow me for using this example, but you will be training the kinematics along those kinds of momentary but ever-present types of disturbances. That's my theory. Well, based on Breitenberg's theory and everybody else on whose shoulder they stand upon. But I think that this is the consequence. If you put an olive like this in a cerebellum like what we have, we're going to be learning unsupervised features of motor control.
Okay, thanks. That's quite clear. Very interesting presentation.
No, thank you.
I have a question.
Please, Julia.
Can you go back to the slides with the coherence that is lost?
Yes. So this one?
Yes. So what like the results are that chimera exists, right? And so that you are losing coherence, so the clusters are like destroying and then recombining, right? And so so this suggests that the most likely model is the one with bridges.
Yeah, absolutely. Actually, let me.
It was just to know if I was right. So
You're very right. I should actually let me put it here. So, so most. Thank you.
Okay, because you are finding a relation to clusters that are far apart. This is what makes you think that, right?
Yeah, I mean the fact that the clusters can be joined together also spontaneously, I mean, depending on their kinship. I mean, surely I am spreading these cells in the model randomly. So I take a subset of cells, right? So I take a subset of cells that have some sort of oscillatory behavior. I'm not having any way in which I'm putting like the similar cells together. It's salt and pepper for all the cells. So some similarity is going to come from the haphazard way in which cells that are neighbors have particular oscillatory frequencies. The bridges can convey information about phase and along, across the entire olive. So the idea is that only with bridges you can do this. If you did with homogeneous, you could produce a wave that would create this correlation, but you will have to disturb the phase of all intermediate groups, obviously. And so you will only have one mega phase that your body can obey. I mean, people talk about the timing function, timing theory of the cerebellum. It's not a bad, it's definitely not a bad name. It has been powerfully misinterpreted, but the state variables that are interesting here are the phase of the groups. And so the timing is timing as a function of the perturbation plus the ongoing oscillation. The ongoing oscillation can be very readily perturbed. I actually haven't shown any data to this. In our paper, the quasi-periodic rhythms of the inferior olive, we've shown how we can perturb the phase in a variety of cells in the entire parameter space.
Okay, thank you.
Of course. Yeah, the thing that we still need to do, Julia, I mean, here is to bootstrap over multiple arrangements of this salt and pepper. What I showed you here, I run maybe 3-4 networks. This is MATLAB code, the GPU MATLAB code. We can run many more networks, but with my code, we reach a limit. And so this is why we got a grant, is because we need to do this faster. And so, yeah, this is the future.
Thank you.
Georgie, how do you feel? You heard multiple cerebellar stories before, so I'm just curious how it comes together.
I feel very excited, actually, and this finding out the chimera states is really exciting, I have to say.
It's fun, huh?
Yeah. Really fun.
So I'm really curious, because if you then have the cerebral cortex playing together, right? I mean, like in the context of our paper, we have, potentially unsupervised way of binding functional groups. And I think that's pretty cool. And so this is what's missing in AI, right? All of the intrinsic dynamics. These cells are all pumping their own activity, and they have their spontaneous, which measures the body, but it's all very spontaneous and different time bases, not synchronous. It's better. Echo-state networks have the problem that they cannot handle phase very well. So liquid state machines either. I mean, they're terrible to handling phase because it's always linear. They're decoding, and so obviously it's going to take time for whatever to change to propagate through the network.
Yeah.
And so with these systems, I think we can propagate changes faster. Yeah, you mentioned, so people are potentially interested in internships, and so there are internship possibilities in my lab. I can also produce. I'll send you a link later to our tentative projects list. Others are possible.
Okay, we can put it on Canvas. Once you send it to me, I can put it on Canvas, and then everybody can take a look at the different positions.
Perfect.
Okay, so any more questions?